Okay, so now that you have watched the overview translation video, we're going to go back to the microbial genetics PowerPoint and talk a little bit more about the details of this process. So the overall goal of translation is to read that messenger RNA that we made during transcription and make proteins. So translation is also called protein synthesis. And we call this process translation because we're literally having to translate between two languages, the nucleic acid language of RNA and the protein language, right? And so that's where we get the name translation, because we have to translate between the two. So we have three key players involved in translation. The first is the messenger RNA. The second is a transfer RNA. And the third key player is the ribosome. So remember, the ribosome has two separate components that will eventually come together to form the intact and functional ribosome. The structure of the transfer RNA is also going to be important. So let's take a look at that. So with this is a single-stranded RNA molecule that has folded on itself. Again, you know it's RNA because it has uracils in it. And it's a single strand running 5' to 3'. And it's called a transfer RNA because it is going to be what brings the amino acids from their place in the cell to the ribosome to actually build the protein. So the T for transfer is because it's transferring amino acids to the ribosome. Okay? So look at the image here. Notice at the very top of the image, but it's the 3' end, it's an amino acid attachment site. That's where the transfer RNA is going to have an amino acid attached to it. So let's pretend it has the amino acid proline. So that tRNA is going to be specific for the amino acid proline, okay? And whenever proline is needed, it's going to bring that proline to the ribosome. It's kind of like a little Uber, okay? Now, how does it know when proline is needed? Well, look at the very bottom of the structure. It has an anti-codon. So the word codon should be familiar to you. Remember, we divided the messenger RNA into sets of 3, and each of those sets of 3 is a codon. Well, this is a set of 3 nucleotides that are going to be opposite to the codon on the messenger RNA. So we call it anti-codon because anti means opposite. So if the anti-codon is UAC, then the codon that it will recognize is going to be opposite of that. So AUG. Okay. Now, hopefully from the other video, you remember that AUG actually brings the amino acid methionine. With the caption here, I just couldn't see what was down below, so I picked proline randomly. But technically, this tRNA would be for methionine because that's the codon on the messenger RNA that it would recognize. Okay, so it has an anti-codon on the bottom of the structure, and then it has an amino acid attachment site at the 3' end in order to hold on to the amino acid that it needs to deliver. The other key player is going to be the ribosome. And so once translation gets initiated, the first transfer RNA is going to bind to the ribosome. So remember, the start codon on the messenger RNA is AUG. That means our first amino acid should always be methionine because that's what AUG codes for. So the tRNA carrying methionine is going to bind to one of the pockets in the ribosome, specifically that middle pocket there. And then what happens is the next amino acid that we need, its tRNA comes and drops it off in that A site, okay? Eventually, the amino acids will be joined together, okay? And then this process will continue. The next tRNA will come in, connect the amino acids, and so on and so forth, okay? And I'll show you more details of that process in a couple of slides, okay? So at this point, you're hearing a lot of words: amino acid, anticodon, codon, right? Lots of terms. So let's kind of think back to what we did in the overview video. So we're going to have the codon wheel, and I actually provide it on this particular slide. So what I've given you, the EX means example. So you'll notice this is RNA nucleotides because there's uracil. And you are going to write down what amino acids are needed. Okay, so go ahead and practice that really quick. Go ahead and push pause and write it out. You might be expected to do something like this on the exam. All right, so looking at the codon wheel, AUG gives us methionine. Remember, your first amino acid should always be methionine. So that's the code MET. Your second codon is CCA. So if you use the codon wheel, CCA gives the amino acid proline. The third codon, GAA, gives the amino acid glutamine, which is the code GLU. The fourth codon there, UCC, gives the amino acid serine, which has the code SER. And then the last and final codon, also called the stop codon, gives you the little solid circle, which means stop. Again, there is no amino acid called stop, so you wouldn't write anything. Okay? So what you notice here is that in order to build this particular protein, we need methionine first. And then we need proline, and only proline, to come in second. Then we need glutamine, and only glutamine, to be third. So it's important that the correct amino acid is brought to the correct, it's important that the correct amino acid is brought to the ribosome at the correct time so we can build the correct polypeptide chain, which will eventually fold into the proper protein. Okay, so it's going to be the transfer RNA's job to bring the correct amino acid to the correct spot. Because how you can think of it is that each tRNA is going to be specific for one amino acid. Okay, so when this ribosome needs methionine, it's kind of like calling a cab. It's like, hey, I need methionine. And the transfer RNA that's attached to methionine will come to the ribosome and drop it off. The ribosome then says, okay, now I need proline. So then a transfer RNA in the cell that's attached to proline will come to the ribosome and drop it off. And then the ribosome says, hey, I need glycine. So then a tRNA in the cell that's holding on to glycine will come to the ribosome and drop it off. Okay. Again, that's why the tRNAs are called transfer RNAs. It is transferring the amino acid from somewhere else in the cell to the ribosome at the proper time. Okay? All right, so let's go ahead and end this video because I just wanted to introduce the key players so you can kind of see what each of their role is. So essentially, the messenger RNA, which is made up of the codons, that's the instructions. That's what's saying, hey, I need methionine first. I need, valine second, serine third, whatever, okay? That's the messenger RNA. It is the message that has the instructions. Then the transfer RNA is going to bring the appropriate amino acid to the appropriate spot at the appropriate time. The ribosome is literally just the location where all of this is happening. Okay, it allows the transfer RNA to attach so that it can drop off its amino acid, attach its amino acid to the others, and then leave. So that tRNA can go and find a new amino acid to hold on to. Okay. All right. So let me know if you have any questions about these key plays.